The neuron simulation is a valuable tool for students to understand the complex movements of sodium and potassium ions within a neuron. It allows students to observe and explore the movement of these ions across the cell membrane, which is essential for the transmission of signals within the nervous system. In this virtual simulation, we will discuss the importance of understanding these movements and how the neuron simulation can aid in this process. The cell membrane of a neuron is a highly selective barrier that regulates the movement of ions in and out of the cell. This process is crucial for the neuron to function properly and to communicate with other neurons. At rest, the neuron has a negative charge inside and a positive charge outside, creating an electric potential difference across the membrane. This is known as the resting potential. The neuron simulation allows students to observe the movement of sodium and potassium ions when the neuron is at rest. At this stage, the cell membrane has specific ion channels that are responsible for allowing only certain ions to pass through. Students can see that potassium ions, which are positively charged, move out of the cell due to the concentration gradient. On the other hand, sodium ions, which are also positively charged, are actively transported out of the cell by the sodium-potassium pump. This process requires energy in the form of adenosine triphosphate, ATP. As students stimulate the neuron in the simulation, they can observe changes in the movement of ions. When the neuron is stimulated, the membrane potential becomes less negative, leading to a decrease in the electric potential difference across the membrane. This change in potential triggers the opening of sodium channels, causing an influx of sodium ions into the cell. This influx of positively charged ions causes a reversal in the charge of the membrane, creating a brief positive spike in the membrane potential. This spike is known as the action potential and is essential for the transmission of signals between neurons. Moreover, the neuron simulation also allows students to explore the concept of refractory period. After an action potential, the membrane potential needs to return to its resting state before another stimulus can trigger another action potential. This is due to the inactivation of sodium channels and the activation of potassium channels, which work together to restore the resting potential. By using the neuron simulation, students can explore and understand the complex movements of sodium and potassium ions within a neuron. This visual aid enhances their understanding of the physiological processes involved in signal transmission and helps them grasp the concept of resting and action potentials. It also allows for experimentation and manipulation of different variables, giving students a hands-on learning experience. In addition to understanding the basic functions of a neuron, the neuron simulation can also be used to explore neurological disorders that are caused by abnormalities in ion movement. For example, students can observe the effects of blocking sodium channels, which can lead to conditions such as epilepsy.